was discussing a little bit before, and this is actually a sort of old project for me, but it is the first time that I'm actually presenting it with the new data that we generated. So I'm very excited. And yeah, it deals with certain things. So we're dealing with innate immune memory and microglia. And I did put, because I wasn't exactly sure of the audience, I did put maybe a bit extensive introduction, but bear with me. We're getting also to the epigenetic part later. But before I start with the data, I would like to acknowledge the people that have been mainly involved in the work, and they are a few PhD students, so they are indicated here. So Chao Ming was one of the students that was really driving the project together with Marissa initially, and then Laura took over with Antonio to sort of bring it home, and I didn't want to start without acknowledging them. Right, so we're talking about microglia today, and they are one of the 4 major cell types in the central nervous system. Right, we have the neurons that actually form the signaling network in the CNS, and that control all our muscles and organs. And then we have the group of cells that we call glia, and there's sort of 3 flavors of those. We have astrocytes that sort of generate a kind of scaffold for the neurons, but are also important in signaling and in part through formation of the tripartite synapse. And they are very critical components of the blood-brain barrier. We have oligodendrocytes, which are the cells that actually myelinate the axons, thereby enabling saltatory conduction and also in that way contributing to neuronal survival. And then we have my sort of favorite glia, the microglia. And they are a little bit different because they are actually macrophages that live in the CNS parenchyma. And as you can see here in this image of an immunohistochemistry against IBA1, they are pretty evenly distributed throughout the parenchyma, and they are thought to, in that way, have neural supportive function. So microglia were discovered a long time ago, a little bit over 100 years, by Pio del Rio Ortega, and he actually already recognized that, right? So he drew these many different shapes and sizes of microglia. So he already recognized their sort of heterogeneity in morphology, right? That is one of the features of microglia, this great plasticity that they have. And microglia, in a way, are also a little bit special in the brain, as I mentioned, because they are macrophages, so they don't derive from the brain tissue that arises during development, but they actually derive from outside of the embryo. At least in mouse, we know this for sure. Right? This was a very nice study in published in 2010, where they did a sort of LAC-Z activation pulse at very early embryonic stages, labeling all the hematopoietic cells blue. And then what you can see is that over time, so around 2 days later, these blue cells are now in the brain, where they migrated just in time for the closure of the blood-brain barrier, and these microglia live in the parenchyma, and they are under normal conditions not replaced by cells from the blood. And what has been done later, of what people have discovered later, so this was a paper from 2016, where they analyzed microglia during development in mice, and it was actually shown that microglia during development actually obtain different types of functions, right? So initially they are very much involved with cell cycle, that then shifts to gene expression profile, suggestive of more functions in synaptic pruning, and then in mice late during embryonic development and early after birth, they shift to this more immune surveillant phenotype that is very critical also for normal brain function. Right, so that then in the adult, they can exert their functions, as I explained with this slide also previously. Right, and just right, one of the things that they do, and I included these movies because I'm a huge fan of them, because I think they illustrate so well what microglia do. So this is a movie where you see GFP expressed in microglia, so everything that is white is microglia. And this is under normal conditions in a living mouse. right? And what you see is that they have these branches that they actually use a lot all the time to move around and to really try to sense their environment. And then as soon as something happens in the environment, which will be here a laser ablation, so there's a white dot here, which is laser-induced damage. And then what I hope you appreciate is that these microglia respond to this damage really fast. And in responding to the damage, they migrate, they change their morphology. So they are really plastic cells in that way. And that heterogeneity is also in the movie. I think it was obvious, but that you can also see that by looking at the immunohistochemistry. And this is examples of human brains where microglia are labeled again with IBA1, producing this, right, labeling microglia in brown. And you can, I hope, appreciate that depending on, right, the image that you look at. So this would be homeostatic microglia in a healthy brain. But with aging or with disease, we see that microglia really change their shape.
Can I ask you something, Susanne?
Yes, of course.
Do they actually touch the neurons, or do they have? I saw that you showed a wonderful video that attacks the area. So yeah. Do they do phagocytosis? Do they actually? How do they function? Will you tell us later?
Yeah, some of it is coming later also. Yeah, but just to give you an answer. So yes, they definitely do touch neurons, and one of the right. So when there is a dying neuron, they will phagocytose this, but they don't only do that to dying neurons, but also in normal healthy conditions. They contribute to synaptic pruning. So really to to organize the network. So they are actually in touch with neurons a lot.
Do they condition the neurons? Do they make them healthy by touching them and providing something?
Yeah, that is the current hypothesis, that under homeostatic conditions, this is what they do. They also produce neurotrophic factors. So, for example, BDNF is produced by microglia. Yeah.
Thank you so much. Thank you.
Supposed to keep neurons happy and healthy. Yeah. Right, and so how do they do this? So this is a paper that was published by the lab that I am in, but not with me because it was before I came. But they were the first to sequence, one of the first to sequence human microglia isolated from fresh tissue. And they actually identified that on human microglia, there's a lot of receptors expressed, right, that are indicated here in the slide, but I will not go in detail, but they express many receptors that they actually use to pick up on these signals that come from either dead or dying cells, or from bacteria or viruses or other things that are not supposed to happen. And then because they have all these receptors, they can respond to that very rapidly. And I have shown you this. So this was for mouse development. And we actually published 2 years ago a paper where we looked at the development of microglia in human fetuses. And what we actually discovered is that they follow a similar developmental program compared to mouse. So this is the graphical abstract of the paper. Right, so you can see what we did. We actually isolated microglia from elective abortion material, and we isolated microglia by FACS and did single-cell gene expression analysis. And then what we found is that in human, so the program they follow is pretty similar, but the timing is a bit different, because in human already within the first trimester, basically, they are ready for this immune sensing function, which in mouse was a little bit later during development. And one of the implications that we think this has is that, right, so when a pregnant mother is exposed to, right, a very strong infection, this actually affects the microglia in the developing fetus. And this is one of the reasons that, right, could result in perturbed CNS development, which is something that there is a lot of correlative evidence that if you have infections during pregnancy, for example, the child has a higher chance of developing autism after birth. So there seems to be really this connection, right, between experiences and functional outcome. And that is what I try to indicate here. So what I have shown you, I hope, is that microglia exist really in a plethora of shapes. And to be fair, I think the actual connection between phenotype and function is still unclear, right? We have, right, hypotheses, but I think there's no definitive proof. And also that for microglia, really these previous inflammatory experiences seem to matter. And that was from the fetal human study hypothesis. But what we know from other work that other people have done and that we have done is that this is actually fact that previous inflammatory experience in microglia matters. And this is regulated through or in a process that is called innate immune memory. So what do I mean by that? So innate immune memory is actually also a relatively recent concept. So it was first described by Nitea around 2016. So this is a figure from a review that I took, right? And I think we all know this classical immunological memory of B and T cells, right? Where there is infection and then there's genomic rearrangements, clonal expansion, and then, right, forever we have this sort of immunological memory. But what they showed is that also innate immune cells, through a process that is not dealing with genomic alterations, or at least not in the changes in the DNA code, but there can actually be this trained immunity also in innate immune cells, where a first infection gives a particular immune response that actually protects the organism from secondary responses, right? So that there can be an enhanced secondary response that can prevent death, for example, in case of sepsis type infections. And this is, I mentioned, so this is organized genetically, whereas trained immunity in macrophages in the periphery is organized through epigenetic reprogramming and also metabolic changes. And this is also true for microglia. So that initial finding was done in peripheral macrophages, but people have also shown that this is true in microglia, where a peripheral immune stimulation, in this case, there is here a mouse that was IP injected with LPS at least 1 time, but right in the graph there is indicated how often, right, where this peripheral immune stimulation actually evokes immune memory in microglia. And how do we know that? So for example here, right before the injection, the microglia were homeostatic. Giving a 1-time LPS injection to mice results in a state of microglia that is referred to as primed, that then, right, in case an additional LPS challenge is given, this leads to hugely increased immune response. So as here exemplified by, for example, increased expression of IL-1 beta and also increased glycolysis. While on the other hand, we can also have a setting where injection of LPS leads to sort of desensitized state of microglia, where a next challenge actually results in a dampened immune response. And this is something that in the lab we measure a lot with, right, with injecting with LPS, but also in a very nice paper in 2018, the group of Jonas Neyer showed that this innate immune memory in microglia actually has neurological consequences. So this is some data from their paper where they show that right, injecting mice with LPS intraperitoneally once or 4 times results in a different training. And then in this particular case, they looked in a mouse model for beta amyloid, so for Alzheimer's disease, the APP23 mouse. And what they saw is that if they injected the mice with 4 times LPS prior to normal onset of beta amyloid deposition, they could see that there was significantly decreased deposition of beta amyloid plaques in these animals, which is probably improving their disease. On the other hand, also, they looked in an ischemia model, and here 1 day post-ischemia, so they looked at sort of the neuronal damage and as well microglia activation. And 1 day after ischemia, there's no effect. But then after a week, it appears that when mice were pre-treated with 4 injections of LPS, actually there is less microglial activation and also a smaller area of the neurons is damaged. Also, again, suggesting that there is really, this immune memory has functional consequences for the microglia and for the animals. And then, so there was a lot of fuss in the field, I would say, about, how should we call these type of things? And I think this is what we ended up with, where we have these 2 opposite responses. So we have this enhanced response that we would refer to as immune training. And this can be established by stimulating microglia that are in a primed state with a particular challenge. And what we would then see is that if this is the normal response to an immune insult, right, when microglia are in primed state, they will give this enhanced response. It can be either immediate or also later, whereas this immune tolerance, the microglia are in a desensitized state, and that means that after the initial desensitizing stimulus, the subsequent stimuli will be dampened, right? So immune training and immune tolerance. Why is this relevant? So I have shown you some of these images before, and I just wanted to highlight again that normal microglia have a particular morphology that is characterized by these long, thin branches and a small cell body, whereas when microglia are aged or diseased, they really have a different shape and probably also different function. And this microglia dysfunction is actually associated with, well, I made a small list here, but I think it's pretty much all neurological disorders. So Alzheimer's disease is very well known, but also multiple sclerosis, delirium, OCD, pain. So there's many things that are associated with microglia dysfunction. And one of the questions that we sort of at the start of this project tried to address was, what are the gene expression signatures and functions that are really associated with these different microglia phenotypes? And also, can we identify the epigenetic characteristics that underlie these gene expression signatures? With ultimately the goal to sort of figure out how they then contribute to future responses of microglia. And to do that, we used these 2 models, mouse models that we have for sort of representing the 2 sides of the spectrum, so immune tolerance and immune training. And I will go into the immune tolerance first. And for that, we use this endotoxin tolerance model where we inject mice repeatedly with lipopolysaccharide in their abdomen. Right, and what happens when we do this is that, so normally the mice before challenge, they have these highly ramified microglia. We give them an LPS challenge, they temporarily change phenotype, they start expressing inflammatory markers, and then over time, the inflammation is resolved, and also the morphology goes back to sort of normal. But what we know is that, so they, in terms of shape, don't look so different. But what we know is that when we have challenged mice with LPS, and we challenge them again, actually, the microglia become hyperreactive, so they cannot so well reach this reactive state anymore. Right. So how do we do that? How did we approach this? So what we did is that we took young adult mice, we injected them with LPS IP. We waited for 4 weeks to let also the peripheral effects resolve, and then we waited for 3 hours after the second challenge to terminate the animals, isolate the brain, and then through FACS sorting, we capture the microglia by using CD45, CD11B, and LY6E as a negative marker. And then we had 4 experimental groups in this case. So we had mice that were controls, so twice stimulated with PBS. We had mice that received LPS first and PBS second, acutely stimulated microglia, so PBS, and then second LPS, and repeatedly stimulated microglia. And what happens when you do this? So this is actually an old experiment that was published by a previous PhD student, Van der Schaafsma. And what he showed is that looking at RNA expression of IL-1 beta and TNF alpha, in this case, that when you when you inject IP, microglia actually start to express both IL-1 beta and TNF alpha. And if you give them a second injection, this response is reduced, as you can see here by this difference. And this is reduced when the interval between the LPSs is 2 weeks, but it is maintained for a very long time, because also when there's 33 weeks in between the first and the second LPS shot, we still see this tolerance. And not only does it work in this way, but I also in my introduction refer to this a little bit. It also works if you inject a pregnant female with LPS first. You let the mice be born, and then when they are around 5 weeks old, you inject the pups with LPS. Then also what we see is that we see this dampened expression of the pro-inflammatory markers, IL-1 beta, TNF alpha, and in this case also IL-6 was measured. Right, so this really seems to be, right, this pre-exposure to LPS really results in a long-term effect on these immune genes that I showed you before. But now what we wanted to do was look at this genome-wide. Right, so again, we used the same setup, but in this case, we then went for RNA sequencing. And what we found was this. So here on the left, you see a 4-way plot where we plotted the expression of the acute response, so comparing the control group with the PL group. And here we expressed the sort of dampened response. So we plotted the control group with the twice LPS treated group. And there's, well, we indicated several genes. They are obviously all immune related. But what you can see is that if you compare right here, the PBS, the control group with the acute, right, we see roughly 1,500 genes being up and down regulated. Whereas if we compare the control group with the double LPS treated, right, the number of genes that are up and down regulated is really dampened, right, suggesting that a subset of these LPS-induced genes is actually not induced so much in our pre-treated microglia. And that is also something that we observed when we analyzed the data in a little bit different way. So here we did this unsupervised hierarchical clustering, and what you can see, and then, right, it identified in this particular case 4 clusters of genes. And then in particular, this clusters 2 and 4, as you might appreciate. So they are genes that are not normally expressed in microglia very abundantly. They are induced by an initial LPS, but then when you give them LPS again, right, the induction is really reduced. We have a group of genes that is normally responsive, and then we have sort of a little bit intermediate group of genes. And when we do a gene ontology analysis of these different clusters, so here the clusters on the side correspond to the clusters indicated down here. And what we see is that, right, so these tolerized clusters, numbers 2 and 4, when you look at the functions that these genes fulfill, they are really mainly related to the immune response, and in particular also the innate immune response. So we identified all these genes that are differentially regulated, but we were also interested in sort of trying to identify what is driving these changes and what are the epigenetic underpinnings of these changes. So besides the RNA sequencing, we also did ATAC sequencing and chromatin IP sequencing, where we particularly targeted several types of histone modifications that I will mention when I will show the data. I changed it and I put it here. So in this particular experiment, we actually looked at the active histone marks, so H3K4 trimethylation and also H3K27 acetylation were included in our analysis. And this is then an example of some of these genes that we knew already to be tolerized. And I plotted here some of the genes as indicated here with the different, so the ATAC sequencing profiles and also the K4 tri and K27 acetylation. And then below here, I am showing you the gene expression data so that you can see that they are really genes that are induced highly in an acute LPS response, but show a dampened expression in when the microglia were tolerant. Right, and I think that is sort of reflected also in when you look at these individual profiles. So for example, IL-1 beta, it's very open in the acute response, and then in the LL condition, right, this is much less the case. But obviously, you cannot look at all the genes individually, so we also here integrated these chromatin data with our RNA sequencing data to really try and make sense of this. And here first I am showing you, right, so again, here we have on the sides, on top is the ATAC data, then in the middle H3K4 trimethylation, and in the bottom H3K27 acetylation. Again, the colors represent the group, so we have the control group and the acute group in this case, and then what is shown here is on the y-axis, the gene expression changes, so that the genes that are more highly expressed in PL compared to PP are here in this area of the graph that is red on the axis. And then on the x-axis, we have plotted the M value, which is a measure for the changes in chromatin that were observed in the different assays. And then what you see is that in this acute response, there's a pretty good correlation between the chromatin openness as measured by ATAC sequencing, but also K4 trimethylation and K27 acetylation surrounding both promoters on the left here and enhancers on the right. Except for this H3K4 trimethylation doesn't correlate so nicely on enhancers, but that I think makes a lot of sense because this is more of a promoter mark and not an enhancer mark. Right, so this seemed to fit really quite well. So what happened when we look at some of the other comparisons? So here we are then looking at the second, so LL compared to mice that received only LPS once and the PBS challenge the second time. Again, here we see that the LPS-induced gene expression significantly correlates with many things. So with ATAC, with K4 trimethylation, and also K27 acetylation, a bit similar to the acute response. But then when we look at the comparison between the acute response and the secondary response, then we start to lose some of our correlations. So the tolerant genes, right, which are the genes that are sort of outside of the middle of the graph, they, after a primary LPS challenge, significantly correlate with enrichment of the ATAC and H3K27 acetylation peaks at the enhancers, but most of the correlations surrounding the promoters are lost, and also K4 trimethylation does not seem to correlate that well anymore, suggesting that the tolerized response of microglia to LPS seems to be mainly enhancer-driven and at least partially explained by a loss of histone marks that are associated with active expression after a secondary LPS challenge. And then the sort of last thing that we also use these data is to sort of try and identify which transcription factors drive these changes. And obviously this is right bioinformatic and predictions, and it's not right. So it's hypothesis. So to look at that, we did this diff-TF analysis, and for that we used the ATAC sequencing data. And then here you can see the transcription factors that are significantly associated with either right, the control, so PP, or the acute, PL. And then there's a few things that that are sort of noticeable. So naive microglia, so they would be in this bluish part of the graph. They are enriched for many known myeloid factors. So MAFB and SPI1 are very well-known microglia transcription factors, so that makes a lot of sense. And also the homeostatic microglia transcription factor SMAD3, which is downstream of TGF-beta signaling, is also enriched in the control situation. And then on the other side of the graph, so in the red part, the LPS response is associated with NF-kappa B, and also immediate early response, interferon, STAT3, general transcriptional activation. So there's many transcription factors that are involved in regulating this LPS response. And then when we look at the tolerized microglia, now appearing here in the middle, what really stood out to us is that here now in the LL part of the graph, we really seem to have lost the activity of the NF-kappa beta transcription factor family, right? They are present here, so NF-kappa B1, REL, REL-B, they are really not present here anymore. And this is, I think, also confirmed by the fact that if we compare directly the acute and the secondary LPS responses, right, then we see that these factors really significantly are associated with the PL condition, but not, and are sort of lost from the LL condition. And on top of that, we find here ZEP1, which is a transcription factor that is associated with suppression of immune active genes that is enriched in our tolerized microglia, which could maybe be involved in sort of keeping these inflammatory genes in check and not so highly expressed. So a short summary of this tolerized part. So, right, we see a long-term change in microglia in response to inflammation. We identified a subset of these immune genes that to be tolerized that are mainly related to the innate immune response. Chromatin at enhancers seems to define this altered LPS response. There's altered regulation of the NF-kappa B transcription factor family. And this is, we don't know, but we have some preliminary data on a few genes, right? There might be also involvement of epigenetic repression through H3K9 methylation that is maybe regulated through G9A, but that is all very preliminary and unsure yet. But we do think that there is also a role for repressive chromatin marks and enzymes and mechanisms. Okay, so that was the endotoxin tolerance. Now we will move to the priming. And for that, we in the lab have been using for a long time this mouse model of accelerated aging. So the mouse is deficient for the ERCC1 gene, which is a gene that is involved in DNA damage repair. Right, so it's a general sort of aging model. So ERCC1 is essential for nucleotide excision repair, and these mice they age really fast, so they have this type of progeria syndrome. And in our lab, in our housing conditions, they reach, well, approximately 3 to 4 months of age, and then they are really, then they don't survive longer. And one of the features that is present in this mouse is that they have really an altered microglia morphology, which is depicted in this slide. So on top, we see different brain areas from a wild type mouse where the microglia are labeled in green. And in the bottom, we see same age or similar age, but then from an ERCC1 hypomorph mouse, where I think it is pretty obvious that the morphology of the microglia has really changed. And not only is the morphology changed, but we also have found that they are hypersensitive, right? So we see these hypertrophic or primed microglia that we know that if we challenge them with, for example, LPS, they give a massively increased inflammatory response, right? And that you can measure. So again, here is an example of a few genes, again, IL-1 beta, for example. And what we see is that in normal mice, this is expressed so low that we don't even see it in this graph. But in the knockout, which are in blue, there is a minor increase of this already under sort of normal conditions. But when you induce with LPS, we see this massive, this really hyper-responsive state of the microglia, and they are really more highly expressing these immune genes. And again, here we had these data from a few genes, but we wanted to look genome-wide. So we used a similar setup as for the tolerance. So here we used wild-type mice and ERCC1 knockout mice. We let them age for approximately 12 weeks. Again, we injected them IP with either PBS or LPS, and then waited 3 hours to terminate and extract microglia. Again, we did our batch of assays, so RNA sequencing and then ATAC and CHIP sequencing. And in this case, we included, besides K4 tri and K27 acetylation, also the K4 monomethylation and K27 trimethylation. So here we had some additional marks that we analyzed. And so this is the RNA sequencing data. Again, a similar setup. We have the different groups. So we have wild type with PBS, knockout with PBS, and then both wild type and knockout with LPS. And then what you see here, when you look at the graph, that it's sort of a bit shifted towards the top left. Many of the genes are a bit shifted towards the top left, which really suggests that they have an increased expression in the knockout compared to the wild type. Right, and again, to give you an idea of what these genes are, so we performed this, clustering analysis. And here we identified, for example, in this area, so clusters 2 and 3 are interesting because they really seem to give this hyperactive response to LPS in the knockout that is absent in wild type animals. And they contain, again, many immune genes, but not only. And so looking at the gene ontology analysis here on the right, so then again, focusing on clusters, these clusters 2 and 3, it really seems to be here also the regulation of immune response, innate immune response, these type of GO terms that pop up in this analysis. Right, so this hyper response, and it makes sense because we gave them LPS, but it really seems to be associated to inflammation. And so again, here we did an integration of our chromatin analysis and gene expression analysis. So I think you will recognize the graphs. So here I'm showing you the ATAC data, and there seems to be a really good correlation overall in all the conditions between the openness of the chromatin and the expression of the genes, which is makes sense, I think. And then also looking at the different histone marks, so K4 mono, K4 tri, K27 acetylation, and K27 trimethylation, right? What we see is, again, there is a pretty nice correlation between these histone marks and where they are supposed to be and the gene expression. And there seems to be a negative correlation with K27 trimethylation, but I think the data points are really very limited, so I'd be very careful to really draw any conclusions, firm conclusions on that. And again, here we identified the transcription factors that are underlying the changes in gene expression. And again, here what we see, so in primed microglia, and that are here, right in the dark green area compared to the wild type, so the control microglia. Again, we lose homeostatic signaling because SMAD3 is clearly identified here on the wild type side and not on the knockout side. And what is gained in the knockout are transcription factors that have many distinct functions attributed to either primed microglia. So for example, lysosomal biogenesis transcription factors, but also inflammatory transcription factors and things related to proliferation. And then when we look at trained microglia versus sort of LPS treated knockout mice, so here wild type LPS and knockout LPS, again with the dark and light colors, what we see in the knockout is that we gain accessible transcription factor motifs for regulators with known roles in LPS response. So for example, NF-kappa B and REL-B, but also IRF factors. IRF7 is, for example, indicated here. IRF8 and 9 are also present. And immediate early response related transcription factors, for example, here FOSB. And again, here also in this set of data, we see that ZEB1 is lost in the knockouts. And this, as I explained in the previous data set, is associated with immune response suppression, suggesting that, right, so this is lost in the knockout, meaning that that is maybe why they have this enhanced immune activity. So what we found was that both in tolerized and trained gene sets, immune system processes were significantly enriched, but we also wanted to really zoom in more closely to the specific genes and processes that were involved in regulating these really opposite hyperreactive and dampened immune responses. So we intersected our different gene sets, which is depicted here on top. And obviously there are genes that are unique to each of the conditions, but there are really also right, quite a few genes that are shared between the different conditions. So now we have, right, a combination of the tolerized experiments and also the ERCC1 knockout experiments. And then the genes that are indicated here in bold. For example, here, these 1006 acute genes, right, been looking at what their function is. So we plotted here a top 5 of GOs, right? They are involved with, well, RNA processing because they have to become very transcriptionally active, but also immune activation was a bit further down, right? But when we then look at what I think are the most interesting gene sets, these 103 and 46, because they are shared between, so they are tolerized in our tolerance experiments and they are trained in our ERCC1 knockout experiments. And when we look at what they are involved in, it's mainly related to this NF-kappa B signaling, which is also right, what came out of our chromatin and transcription factor analysis. So that made a lot of sense to us. But again, here we wanted to zoom in more if we could identify the factors that drive these responses. And to do that, we again went to this DiffTF analysis using the ATAC sequencing data. And what we then found, so what you see here is a heat map where the genes identified in the different comparisons are indicated in columns, and then on each row, right, we have one specific transcription factor. And what you, I hope, can appreciate is that it really seems that these different, so these 4 different responses of microglia, so acute, tolerized, and then primed and trained, right, they really seem to be regulated by discrete groups of transcription factors. And maybe this is why we see these really opposite responses, right? The same genes are affected, but they are in the different experimental setups regulated, seem to be regulated by different transcription factors. So in summary, this is a sort of graphical abstract that we made of the paper. So we had these 2 different mouse models. So we had on this side the tolerance mouse model, where mice received 2 consecutive injections with LPS. On this side, we have the ERCC knockout model resulting in a primed microglia phenotype that we analyzed by these genomics approaches. And what we saw is that in case of microglia desensitization and tolerance, right, what we see, what happens is that in the acute response, NF-kappa B related transcription factors are recruited, immediate early genes, transcription factors are recruited, leading to very high expression of these acute LPS genes. Whereas in for a subset of these genes, at least when you treat mice again with LPS, we see that NF-kappa B is no longer recruited to the chromatin, Instead, ZEB1 might play a role, and this together results in a much dampened response of these gene expression response in these genes. On the other side, in the microglia priming and training, right, in wild type situation, there's not much going on. Then what happens in knockout mouse is that on particular genes, there is already some accumulation of particular transcription factors that when these microglia receive an inflammatory insult, in this case it was LPS, on top of that, NF-kappa B is also recruited together with these immediate early genes and IRFs, and this results in a much higher expression of these trained genes compared to wild type situation. So that was a summary, and right, so that was so we closed the story with that. But I actually think there's a lot of open questions, and this list is in no means conclusive. But I think in terms of tolerance, some of the things that we have been trying to address is how repressive marks are involved, and also which histone modifying enzymes are responsible for making these chromatin-related changes. One of the things I think is that we are trying to address is, right, can these epigenetic marks be erased or restored so that homeostasis can be returned to the microglia? I think we don't know the long-term actually functional consequences for the cells themselves, but also for their CNS environment. And I think also what we don't have evidence for is whether these processes actually also occur in humans. And I think this is a relevant question because also the turnover rate of microglia between mice and human is not exactly the same. So I could imagine that right, the storage of epigenetic information could also be slightly different between mice and humans. And with that, I would again like to show you the people that were involved. And yeah, I'd be very happy to take questions.